Hello and welcome to today's webinar session featuring André Fenton. I'm Greg Bousted from the Simons Foundation hosting today's session. We'll kick things off in just a moment, but first I'd just like to let you all know that you can ask questions at any time during today's presentation. You can just ask them using the text feature to the bottom left-hand corner of your screen. We'll be monitoring those throughout the session, and at the end of today's webinar, we'll get to as many of them as we can. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere, or if you have first obtained express written consent from the presenter to do so. Okay, André Fenton joins us today calling in from New York University, about 10 blocks south of our offices here in New York City. Dr. Fenton is professor of neuroscience at NYU's Center for Neuroscience. His lab studies memory and, in Andre's words, how the expression of knowledge activates information that is relevant without activating what is irrelevant. As part of this work, Dr. Fenton seeks to understand the implications of neural coordination. In particular, he is interested in investigating how neural activity is highly coordinated to be in sync and out of sync depending on different situations, and to understanding the implications of when this neural coordination is disrupted, as seems to be the case in conditions such as schizophrenia, depression, epilepsy, and relevant to today's presentation, autism. We're very grateful he's taken the time to share some of this early work with us today, as well as give us background on his approach. Welcome, Professor Fenton.
Thank you very much, Greg. So I'll begin with really the central question that my laboratory studies. Everyone in my lab studies a very particular problem, but the real umbrella problem that governs everything that we do in the laboratory is really this question. We're interested in how thinking about things, cognitive experience, changes the functions of neural circuits. And today I'm going to talk about how we study that, but in particular how it may have implications for autism, and more specifically we'll be studying an animal model, or I'll relate data on an animal model of Fragile X syndrome. I want to remind some of us what the clinical features of Fragile X syndrome is. It turns out to be one of the most common inherited forms of intellectual disability. It's X-linked. There's a single gene. The gene is silenced. There are a number of CGG repeats in the gene that causes it to be silenced. The gene's name is FMR1. This slide shows you the prevalence. It's a collection of syndromes. I'm going to focus on the intellectual disability. There's certain rather subtle learning disabilities that ranges to severe intellectual disability. It's most significant in males. And you can recognize in about 40% or so of people with the mutation characteristic features of autism. We will focus again on the trying to explain how this single mutation leads to these kinds of changes that are recognized at the level of cognition. The next slide here shows what our challenge is. I was going to say goal, but it's truly a challenge. We know what causes this. There's a mutation in the FMR1 gene. Okay. We've, through a lot of work through others in the field, have identified that the function of this gene is in regulating how mRNAs are translated into proteins. And this slide here shows that in panel C, that there's more global protein synthesis in the region of the hippocampus. That's what's focused here, but it's happening throughout the brain. More protein synthesis when this gene is not functional, as in this case, it's been knocked out in a mouse. And that dysregulation of protein synthesis or protein manufacture somehow leads to the inability to make appropriate decisions, the inability to change rules once they have been learned, what can be called in gross terms cognitive inflexibility. And what we'd really like to understand is how we go from that mutation or loss of the translation regulation to the cognitive inflexibility. There are a lot of steps there represented by these three simple dots, and our goal is to try and explain them in one way or another. And the way that we have chosen is to recognize that this is a systems problem. I'm going to change slides here. The next slide shows what I call the circle of biological being, sort of tongue-in-cheek. If you look here at the individual level and at the level of behavior, individuals obviously function within societies amongst other individuals, and the society or the groups that that individual participates with obviously influences what the individual's behavior would be. That individual's behavior is influenced and determined in large part by its brain. And what the point of having these arrows go in both directions is all of the causality, the influence of the brain on the behavior, and the behavior on the brain goes both ways. And that's what makes trying to understand how you go from a genetic mutation and dysfunctional translation over here to abnormal behavior, either through this series of pathways or all the arrows that aren't shown here, difficult. And it makes trying to study this problem particularly challenging because the behaviors that we are particularly interested in happen to be behaviors of cognition. And what do I mean by that? When we think about cognition, it's a fuzzy term, but I'm going to talk about it in a rather particular way. What we refer to as cognitive is all those mental abilities, those processes that are related to, to knowledge in general. And the important thing to recognize is that brains in general do things that are mental and related to knowledge. And how are they distinct in any way from things that are in the external world or more attributable to the senses? As this cartoon shows, there's information here from our five senses that enter the brain, and while there's cognitive information processing that may occur on the information collected from the external world, what's particularly interesting is this stuff that happens internally with that information. And we in the laboratory are very interested in those internal processes. And one of the reasons we have chosen to study the brain in the way we have is because we have found a model system that gives us access to information that almost surely has to be generated internally and can't be attributed to any particular sense organ, what I would call a sensory epithelium. It's not connected to the skin, to touch, to vision, to sight, to sound, to smell, or any sense in particular, and that thing is going to be our internal understanding or representation of space. And the reason for focusing on this is because it is the kind of information that we can verify experimentally, but we also know either philosophically or just through a little bit of introspection that that kind of information has to have a computational origin. It has to have been computed internally rather than simply represented from the outside. And so we're going to study a particular kind of neural activity that's cartooned in this picture. It's what I will call neural coordination, and that's a term that describes a set of processes that organize, in particular, the timing of neural activity. And it's the timing of neural activity with respect to other neurons. So how one neuron discharges with respect to other features and other discharge of other neurons. And in particular, it's this coordination that we're interested in independently of the discharge or the neural activity that's characteristic of an individual cell or of an individual population of cells. This coordination is really focused on the timing and the changing of timing of neural activity. And it's our hypothesis, and the focus of the work that we do, that these kinds of processes, these timing processes, are really essential for the information processing and the neural representations that we'll study. And what's shown here is a slice through the hippocampus. Superimposed are the local field potential oscillations and the rhythms that we can study at these different locations along the hippocampal axis. I'll unpack some of these figures and show them to you later. Here's another way of looking at the same data as events of these oscillations. And down here is a figure from a paper demonstrating that the spiking activity of individual neurons happen to be organized by the phase of the ongoing local field potential oscillations. And we will try and study these interactions at all levels throughout the talk. Okay, so let's really try and begin. So we're interested in cognitive experience, and I'm going to try and frame for you how we approach that and what the cognitive variables are that we can study in the laboratory. We're going to focus on a behavior or an observation that you can infer. It's known as cognitive control, at least I'll call it that. It's also known as executive function. If we were psychologists, we could fret about how these things are distinct. But for my purposes, as an empiricist, we can recognize cognitive control as really that ability to coordinate the use of information from multiple sources, at least more than one, and that optimization or coordination is typically used to direct actions and to direct choices so that you get optimal outcomes. As an aside, cognitive control has some value in studying in its basic form and its translational utility because it's impaired in many forms of mental illness and is actually a major therapeutic target. So if you could improve cognitive control in various patient populations as shown here, including ASD, schizophrenia, depression, etc., you can improve functional outcomes clinically. Okay, so a classic way of looking at cognitive control or measuring it in the clinical laboratory would be the Stroop test. I would give you an instruction like this, say the color and please ignore the meaning, and I could present to you characters like this, organized RED or BLUE in different colors, and you should be able to recognize that there's at least two interpretations of each one of these words. One is the meaning, and the other is the actual color of the letters. And given that they can be presented here in a congruent manner, this presents very little challenge. However, if it was presented in this color incongruent way, the word RED, spelled in blue ink or blue photons, that presents a challenge. Now you have to actually willfully, and if one ever does this test, you can actually feel the cognitive effort that's required to actually focus and ignore the meaning of the word red when you're confronted with RED written in blue. So we're very interested in this kind of process and how this kind of process can be accomplished by a neural system. And so we've decided and tried to work out how we could study something at least analogous to this in rodents in the laboratory. And we took advantage of the ability of animals to organize where they are in space, and the fact that space can be defined arbitrarily in multiple spatial reference frames. So this cartoon shows that a rat can try and locate itself in one instance by recognizing where it happens to be within the space defined by the enclosing room, or in another instance, in fact, the very next moment, where it happens to be with respect to the substrate that it happens to be on. And I'll show the next movie. And if we create a situation where what we do is we put an animal in an environment where we can have the floor rotate and the animal can look very much like being on a carousel, out into the world and recognize where it happens to be with respect to the contours of the room, as well as where it happens to be with respect to the floor. You can imagine it's like being on a carousel where you can localize yourself somewhere in the amusement park versus somewhere on the carousel itself next to the golden lion or the brown horsey, that kind of thing. And this, when implemented for rats, looks something like this. The red mark here shows where the what's called a shock zone is defined, and if the animal is detected in entering that shock zone, then the computer will electrify the floor, and the animal won't like that. Oh, I just lost the video. The animal will try and avoid that. It will also try to avoid being in the rotating part of the floor. And if you think about that, it's the same shock that it's going to receive for entering a particular part of the floor or a particular part of the room. But the animal has to make a decision in how to interpret that shock. One reason for getting shocked would be because it has entered a particular part of the room. The other reason for being shocked would be because it's entered a particular part of the rotating floor, and it has to make the appropriate association. And if it doesn't, then it will be very difficult to avoid shock, and it will appear to be being shocked randomly. However, as you can see here, rats learn to do this, and I'll demonstrate later. They learn to do this very rapidly. This isn't very difficult at all for them. However, it does depend on an intact hippocampus, and it requires hippocampal plasticity for animals to be able to remember this over periods of time. So this is the kind of task we'll be using, and we have various variants of this that we'll use so that we can create a cognitive challenge for animals. So now that we have at least a task that we can give animals to do, how are we going to measure these cognition-related operations of the hippocampus for our neural circuit? So here I wanted to point out and give a praise to John O'Keefe and the Mosers in Norway. John O'Keefe is at University College London, but he actually hails from New York, where I am. He discovered place cells in 1971. The Mosers discovered grid cells in 2005 as well, and they've described other cells in the hippocampus and related areas that seem to be related to the animal's notion of where it happens to be. And for this, they were awarded very happily the Nobel Prize. I'm going to show you a video of a place cell that I recorded when I was a graduate student. Hopefully this will load. It's basically an animal. It's a rat. We're able to, we've implanted electrodes into its hippocampus, so we're able to record the electrical activity of individual neurons. You'll be able to hear one of those neurons, I hope, as it discharges. And if you pay attention, as John O'Keefe and others have, to where the animal is when this cell discharges, you'll be able to recognize that it's discharging with respect to a particular part of the room, a particular part of the cylinder that you'll be able to see. And by collecting this kind of information and looking at the properties of these kinds of cells, one is able to recognize, let's hope this plays, that there are cells in the brain that are capable of signaling the animal's location in space. Anyway, this would be boring. Let me go on to watch the next slide. The slide up here, let me point to it, okay, is, or this picture is a representation of where the activity of the rat, as we were just not watching, but I had hoped we could watch, where that cell discharged as the animal moved through this space. So this is over a 15 or so minute period of time. This cell is mostly quiet everywhere in this cylinder. It's about 1 meter in diameter, but it discharges over here most of the time, a little bit over here. And here is an ensemble or organization of 5 cells recorded simultaneously from the hippocampus, and what you can recognize is one of the cells has what's called a place field over here, another one over here. Each cell has a characteristic part of the world in which it will discharge, and it will discharge as the animal roams around and walks through this space. And if you take the activity of these cells collectively, I want to point out that it's only a subset of the cells that actually are active. This cell and this cell fire no action potentials at all. These two fire very few. This one's diffuse. This is characteristic of what one observes in the hippocampus. If you take the collective activity of these cells, it's easy to imagine that they make a map, a representation of this overall environment. And we're very interested in studying that representation. So how do we think about this representation? What does it actually mean to say a representation, and how is it that the brain might actually carry information in the activity of these cells? Well, there are two basic ways to think about any neural representation. One viewpoint is what is called a dedicated or cardinal coding point of view, and an example of that is your typical stoplight or typical traffic light, excuse me. If you look here, analogous to a single neuron, when this green light goes on, it's unambiguous. It's very clear what it's signaling. There's no mistaking it. It means you can go. When a different light goes on, let's say the red one, it's unambiguous. It's very clear what's being signaled. It means you can stop or you need to stop. Each light is devoted or dedicated to signaling a particular kind of information. On the other hand, you can represent the same kind of information, go as well as stop, in an across cell, or I'll call a cell assembly or ensemble coding point of view. Here, there's multiple light elements that go into making, in particular, a pattern. A pattern of on and off lights is what carries the information. No particular light turns out to be important. No particular light is unique or special for making this signal, at least in principle. And perhaps the most common form of this is to look at a stadium jumbotron, the kind of thing you might find at a football game. And this will play. I have a fairly exciting movie of the game over presented here, and I think that's a good analogy for how we'll think about these cells from the point of view of an ensemble code. Okay, as you can see here, you can recognize the pattern game over. No particular light is necessary for displaying this. Each light can be made analogous, if you will, to a particular neuron. And it's the pattern that is communicating the signal here. The temporal organization of the pattern is what makes it recognizable. They don't all have to be on at exactly the same time, but as long as they're approximately coactive, those cells, by analogy cells, those light bulbs show us and are able to communicate a particular pattern. No particular light bulb was crucial to it. And so that's an alternative way of signaling information. And the question is, how should we think about place cells, and how should we think about how place cells are carrying information? And we'll have a very clear answer for that, and we'll talk about that in terms of cognitive control and neural coordination in the discharge of these hippocampal cells. This is the work of a postdoc or two postdocs in my laboratory. When they joined my laboratory, I had them learn how to record place cells, and they conducted the following kind of study. We were interested to understand how these place cells might discharge with respect to the classic environment, which is a cylinder. In our case, it's 76 centimeters in diameter, which is the standard arrangement of things, versus in this larger chamber, which was about a meter, 1.4 by 1.5 meters. And the animals were very familiar. These were rats. They were very familiar with running around the cylinder or running around the chamber for at least a month before we implanted the electrodes. And when we did that, we could record these place cells in a classic way, you'll notice that the individual cells had single places in the environment where they like to discharge, the way these cells are typically described. This is one 15-minute recording followed approximately an hour later by another 15-minute recording, and you can see that the patterns are more or less stable. This is the classic stuff. However, if we let the animal in the intervening time during the 1 hour run around the open chamber, you see a rather different pattern. The first thing you'd see is what's called remapping, which isn't surprising. This has been described since 1987, that the cells will change where they fire from one environment to another. But I think what's more remarkable is that these cells that used to discharge in single locations in a slightly bigger space are now discharging in multiple locations. If we take, for example, this cell number 4, and you try to decode that cell and say, where is the rat from the activity of this cell, it would be rather easy in the cylinder because it fires in a particular single location. Whenever that cell fires, you would guess the rat's somewhere over here near, let's say, 5 o'clock, 4:30 or so. However, when this cell fires in the bigger chamber, it's ambiguous as to where it is. And no matter what you chose, any algorithm you chose to take the average, or you'd have to guess between one location or the other location, you're guaranteed to be wrong at least half the time if you guess, and if you took any average, you'll be somewhere along the line and probably wrong all the time. And so that would be the dedicated coding point of view and interpretation of this cell's activity. However, as an ensemble, if you collected the pattern of activity of these cells all across the space and asked, what is the population pattern like at every location, either in the cylinder or in the chamber, you'd recognize that it's very clearly unique at every location, and there's no difficulty or no ambiguity in decoding where the animal happens to be. And so from henceforth, as we'll talk about these cells, I'd hope that you'd agree that the proper way to think about them is from the viewpoint of a cell assembly or an ensemble code, and a dedicated code would be inappropriate for trying to understand the spatial information in these cells. That's going to be important as we use these cells in the next few studies that I'll talk about and start to study how they discharge with respect to each other. If you look here, I've taken the same data and taken portions of it, and I'm now representing those data not with respect to where the animal is, but with respect to time. So I've shown here a raster of activity of these cells as a function of time. And if you squint, you can almost recognize that the activity, the timing of activity, is different in the cylinder than it is in the chamber, and it's reproducible when returned to the cylinder. And a way to quantify this is to do what we would call this cross-correlation matrix of the activity vectors. What's shown here is simply taking a sliver of time, let's say every 1 minute, in this what I would call a trivial example, and we can count how much activity each cell discharged during that time, as well as during every other time, and we have these descriptions of the collective activity across the population as a vector. And in this correlation matrix, we can simply ask how similar is the activity at this moment, let's say at minute 3 compared to minute 2. Okay? And it seems to be very cell-similar for the first 15 minutes. Then it becomes different for the middle 15 minutes, different to how it was in the first 15 minutes. And then it returned to being very similar in the last 15 minutes to how it was in the first 15 minutes. And during the middle 15 minutes, the activity was also very cell-similar. And this is, in some sense, a trivial example of showing you that if you just looked at the across-cell timing of this discharge, you could recognize that the animal was in either the cylinder or the chamber or returned to the cylinder just as well as you could from the spatial firing patterns. And going forward, what we will start to study is the temporal organization of these cells in order to decode in a very similar way. We're just going to do it on a much finer time scale. And we're going to do this so that we can discover things and the kinds of information that the cells carry as a cell assembly or as an ensemble that you couldn't recognize from these time-averaged firing rate patterns. So to do that, we'll look at the work of two graduate students in my lab. They've both since graduated and have moved on. Edward Kellerman is now in the Czech Republic, just starting his own lab, and Dino Dvorak just finished his PhD with me. And what we decided to do was ask animals to do that task that I don't believe you saw the video of, but this is the task where the animals are walking around a cylinder that happens to rotate once per minute in a room, and it has to know where it is in both the room and know where it is on the cylinder surface in order not to get a mild electric shock. And I'll just walk you through this. So this is a track of the animal during the 15 or 20-minute session. And you can see when the animal entered this part of the room, it received a foot shock, shown here in red circles. But those foot shocks were dispersed in random or unpredictable parts of the arena floor, whereas when the animal entered the particular part of the rotating floor, as this moved around, those shocks collected here, but were dispersed with respect to the room. And this is analogous to having to make sense out of an ambiguous stimulus, for example, the shock, that has two interpretations, one being for being in a part of the room, the other for being in a part of the floor. And so what do we see when we record place cells in this kind of condition? Well, the first thing I'll show you here is, in this case, the arena wasn't rotating, but the animal had learned to make this avoidance. And this is just an example of the kind of thing that I mean when I talk about neural coordination. Here are three cells that were recorded simultaneously. They have very similar time average tuning curves, but if you look at how cell A fires with respect to cell B on a short time scale, the time scale that neurons probably care about each other on, 200 milliseconds, you'll see that cell A and cell B really don't like to fire together exactly the same time. Whereas cell B and cell C, they actually really do like to fire together on the same time scale. And cell A and cell C here, this one and this one, they're willing to fire together, but with something like a 70 or so millisecond offset. I should just point out that the animals aren't changing position on this time scale. They move about 3 or 4 centimeters in approximately 117 or so milliseconds. So they're mostly stationary while these data are being collected, but there's something inherent and reliable about how one cell likes to fire with respect to another. So we can do a little bit better than just describing that. When the animal is on this rotating arena and is avoiding both locations of shock or in the room as well as in the rotating arena, we can observe the activity of a set of cells. I'm showing you here a single cell on the left and another cell on the right. We can observe the activity of those cells in the reference frame of the room as well as the reference frame of the arena. And what's shown here is the activity in black here every 117 milliseconds, how many spikes were discharged, what's the rate as a function of time, okay, for both cells. And what's shown here in red and blue is this calculation. I call it momentary positional information, or IPOS. It's computed in this way. You can think about it as telling you the amount of information about the animal's location in the discharge of that cell, okay, during this particular moment. And you can see here that this cell on the left, when it discharges, tells you very little about where the animal is in the arena, but it tells you quite a lot about where the animal is in the room. The complement is shown in this cell. When it discharges, it tells you a fair bit of information about where the animal is in the arena, very little information about where it happens to be in the room. The good news is this is relatively easy to understand. The animal is aware of where it happens to be in the room, and it's aware of where it happens to be on the arena. The bad news is these are about these are two examples of approximately 4 cells that we've seen in, let's say, 1,000 or so recordings. These are extremely rare. What we typically see is cells that signal on one occasion where the animal is in the room, and on another occasion where the animal is in the arena, and they transition or switch between these two. What's shown here in this plot is the collective information from an ensemble of cells and the difference between the information in the room and the information in the arena. So when the information is positive in red, it means this ensemble preferentially signaled information where the animal was in the room, and in blue preferentially where it was in the arena. And you can see that this system is switching back and forth between signaling locations in both spatial frames. This one is a room preferring overall. This one is an arena preferring. And these just happen to be regular old place cells if you plot their firing fields. We can do a little bit better than that. When a pair of cells signals the same kind of information, they like to fire together within a time window of approximately 25 milliseconds. That's the time window of a gamma cycle, which we'll talk about shortly. If they tend to signal different kinds of information, like room and arena information during this moment, then they tend not to have a preferred relative timing. These are just examples of the coordination, and what we can recognize here is if you plot this is just zooming in on a shorter time scale, and you plot how close the animal is to the room shock zone versus the arena shock zone. When this number is positive, it's closer to the room shock zone. When it's negative, closer to the arena shock zone. You can see that the activity in the hippocampal ensemble tracks very closely on rather sharp time scales, short time scales, where the animal happens to be. And you can see that even closer, if I can go back. Here, as the animal gets closer to the arena shock zone, it's right at the edge. The activity flickers from wrong back into right and settles into the correct reference frame, tracking where the animal happens to be accurately, as if the hippocampus is choosing to represent space most with highest fidelity in the spatial frame that is, if you will, more dangerous, in the spatial frame closest to where the animal needs to actually pay attention. So what I hope to have shown you so far is that place cell discharge is an ensemble code. That discharge is multi-stable. It switches states and is dynamically grouped into these functionally defined groups. Let's move on. What I'm going to show you now is how this activity is organized in the context of ongoing local field potentials. And the best way to describe this is, if those of you who surf will recognize this, those of you like me who are not surfers or can't surf, I'm going to show you a movie. This movie is going to show you that scene where each surfer you can think of as being analogous to a particular neuron, embedded, if you will, in a set of waves. Those waves are synaptic waves, the local field potential. And what you'll recognize is that most of the surfers will feel the presence of the wave. They all go over them. However, there are only these 3 surfers, you'll see them emerging, that caught the wave at the right phase. They're the ones that actually capture the energy or the influence of the wave, by analogy, the synaptic wave, to do some work. For example, in the surfing analogy, to move closer to the shore. So it's the timing of activity or activation, excitation with respect to ongoing neural activity, ongoing synaptic activity measured as local field potentials, that turns out to be important. And one way to describe this is what we call a phase frequency spike plot or phase frequency field coupling. And what's shown here is we've got the LFP. We can filter it for different frequencies shown on the y-axis. And what's shown on the x-axis is the phase of the different waves at these frequencies, what's plotted in Z in the color is the probability of spiking at a particular frequency, at a particular phase. And you can see here that, for example, at theta, this cell likes to fire on the near the troughs of the oscillation. This cell here at this time likes to fire preferentially over at the rising phase of the oscillation, and so on. And these arrangements are different for place cells, for cells that happen to be non-spatial but are pyramidal cells, and interneurons. These cells have different relationships to this, on average, to the local field potentials. And you can see here another set of examples. I won't dwell on this in interest of time, but here's a pair of cells recorded on 2 occasions, and the phase frequency relationship of each cell is characteristic and unique, but also reproducible. And we're very interested in these relationships. So I've described for you a bunch of neural coordination phenomena, the kinds of things that we try and record and decode in order to understand information processing through the hippocampus. And now let's look at how some of these things are dysfunctional or disorganized, discoordinated, as I say, in Fragile X knockout mice. So these mice have, this work has been done by Dino Dvorak, Basma Radwan, and Fraser Sparks. And these mice have been generated and studied very well. David Nelson generated the mice that we'll be studying. Mark Baer and Steve Warren have made major contributions in working out what the consequences at the level of translation of proteins, what the consequences of losses of FMRP, the protein that FMR1 encodes. But we still had that very big gap between the translation dysregulation and what it is that causes these cognitive deficits. And according to the discoordination hypothesis that we've developed and argued over the last several years, is what we would expect is this coordinated hippocampal place cell firing, coordinated with respect to one neuron to another, as well as with respect to the ongoing local field oscillations. These would be abnormal in the FMR1 knockout mice, despite the individual cells and their place cell properties and their unitary properties being relatively normal. So let's take a look. As I think many people who study these mice know, that maintaining the strength of their synapses is actually dysregulated in these animals. That's a fundamental property of neurons. We're going to study this in mice. I'm going to skip over, in the interest of time, the next movie here, which was going to show you mice doing the version of the place avoidance task. And this is something that we did in Woods Hole, where I happen to be the co-director of the Neural Systems and Behavior course, where we teach many of these techniques in an approach, a systems-level approach to studying big problems. With my colleague, Professor Juan Marcos Alarcon, what we did was we trained these animals in the task that I didn't show you, but I'll show you in a moment. After they learned the task, we were able to make hippocampal slices, and we could measure what the strength of the Schaefer collateral synapse was. And one of the things that we've been able to demonstrate is that after training, this synapse becomes stronger. But in the knockout, in the FMR1 null mice, that synapse doesn't become stronger because it's already very strong. You'll notice the scale bar here is twice that as it is in the wild type. If you give a high-frequency stimulus, 100 hertz, you can induce LTP. In the trained animals, that LTP is attenuated. These slices, by the way, were made 24 hours after the training, so these are persistent changes. In the FMR1, you can see that there's rather a large difference in the plasticity that you can induce after training. It's much less than it was before training in the FMR1 knockout mice. So just like others have described in other forms of LTP, we can see an activity or training-induced set of abnormalities in these animals. We'd like to now understand how that translates into abnormal behavior. And so what we've done, what I'm showing you here is a series of place avoidance tasks where the Fragile X model, the knockout mice, look to have pretty normal learning. What's shown here on the y-axis is how many errors or entrances into the shock zone that they make, and it's decreasing rather normally. After a 24-hour delay, they remember rather well. Their inflexibility appears when we change the shock zone from the initial location in the north to the location in the south, and that now requires that the animals who have been well trained and well learned to avoid the north, because they spend a lot of time here in the south, then they become impaired. While wild-type animals, both genotypes are equally disturbed. While wild-type animals rapidly learn to avoid the new shock zone, the FMR1 animals do so much less. They also have an extinction deficit that I won't describe here, but along the same lines. They demonstrate an inability to change what they do with respect to the new information. Just in case you want to see what that looks like, this is an animal that learned to avoid going to a location in the north. It's hesitant to go in there because it has learned to not be there because there was shock, and now it has to learn to stay out of this new shock zone, which is not doing a very good job of because it's just exposed to this for the first time, but it will learn. So when we look at the local field potentials in the wild-type and FMR1 mice along the hippocampal axis here, the stratum pyramidale, stratum radiatum, stratum lacunosum moleculare. Now remember, these are all input-specific regions. If we take the raw local field potential and we filter it for activity in the theta band, that's the slow waves that you see here. Let me get my pointer. These slow waves. And you filter it for activity in the faster gamma bands, that's these fast waves. This is inhibition that is timed by theta, and we know the biophysical origin of that. When you compute this relationship between the amplitude of the fast signals compared to the slow signals, you compute something called the modulation index. This is showing the modulation index in the neural space defined by these different synaptic or input regions, the stratum lacunosum moleculare. That's the direct input from the entorhinal cortex. This is an indirect input from the entorhinal cortex, the perforant path input to the dentate gyrus. And shown here is the direct input to CA1 from the entorhinal cortex, stratum radiatum. You can see that the activity on average as the animals went through pre-training, trial 1, trial 2, trial 3, their retention at 24 hours, their conflict when the shock zone was changed, or extinction when the shock zone was turned off. The knockout animals seem to occupy a different part of that neural space compared to the wild-type animals. And we can demonstrate that that has some relationship to how the animals perform. What we're able to do is we can look at a little more fine scale at the individual oscillations in the ongoing local field potential. We can take individual oscillations here in the LFP represented by a frequency time representation, and we can now study those individual oscillations by a variety of different features so that we can identify them. When we do that and we study how these oscillations appear just before the animal's about to run away from getting shocked, what we can see are recognizable patterns. I won't describe them in any detail for the time being, but we can recognize this kind of pattern across the different locations where the electrodes have been recording, and this pattern describes that the animal is going to successfully avoid, and when the animal fails or is going to make an error, the pattern is very different and very recognizably easily different. And this is, of course, all activity while the animal happens to not be moving, because that's what we selected for, as it approaches the shock zone and is going to run away. That's what wild-type animals look like. If we look, you can see the development of this as the animal is trained. In the control animals, the knockout animals have a more dysregulated pattern. This is, again, all in the timing of these ongoing oscillations within which the spiking is embedded. So let's take a look now at the spiking of these cells of, oh, I just went a little too fast, of place cells. Okay. And so place cell activity is mostly normal in the knockout animals. Here's a set of them recorded while the arena was stationary and then while the arena was rotating from the room frame point of view, and they're mostly messed up in the arena frame, relatively stable. This is the normal pattern. What's interesting, however, is if one looks now at how the cells are discharging one with respect to another at here the 40 millisecond time scale, you can see that the distribution of looking at the pairwise correlations before training and after this active place avoidance training, the wild-type animals seem not to be changed by that. They have a balanced distribution. But the FMR1 knockout animals, they start out already skewed to the right, so they have cells that are more discharged in a coactive way, but training really moves that to the right. Okay, so training is disturbing or changing the discharge in these cells. If you now look at the phase frequency spike field coupling measures, this is a measure of stability. The lower this number, it's the mean squared error across the theta band frequencies here. In the Fragile X, this is notice an error. This is reversed. You can see that the knockout animals are much more stable. They're in fact very rigid in how their spiking is related to the ongoing field potential. And we'll just finish up by summarizing. What I hope I've convinced you of is that cognitive experience is able to change and tune the synaptic communication pathways, demonstrate that this is able to create and, in some sense, even control information flow through the brain. And you can measure this kind of thing in phenomena we call neural coordination. This actually depends on the synaptic functional network, and it itself is changed by cognitive experience and is describing, in some sense, an infrastructure within which spiking occurs, within which information can be processed and used. And it's a discoordination of these kinds of features that can account for the pattern of intact memory and impaired cognitive control, impaired flexibility that we can see in our autism model mice, the FMR1 knockout mice, as well as other animal models that we study that have various kinds of cognitive control deficits. And if there's any really global take-home message, it's really, if you try to understand neural coding and try and decode the activity of neurons, there are many fundamental brain operations like the functional dynamic grouping. These kinds of coordinating operations could not be detected by over-averaging across long periods of time, or in particular recording or even analyzing the discharge of one neuron at a time. You have to look at the population, and you have to look at a minimum the interactions across cells. So with that, I'd like to thank my collaborators. The people who've done most of the work I talked about today are shown here in red. This work, most of the, or all of the Fragile X data collected in the single unit studies was funded by the Simons Foundation, the National Institute of Mental Health, also funded the local field potential work in the Fragile X model, and the National Science Foundation funded a lot of the early work in the development of the ideas about cognitive control and neural coordination. Thank you very much.
Okay, well, great. Thank you so much, Dr. Fenton. That was a really fascinating talk, and it's really neat to see some of that, the early indications of the autism model. I have one question before we get to maybe one or two of the questions from the chat before we end today's session. We hear a lot, of course, in covering autism research, long-standing hypothesis has been about, the balance of excitation, excitation inhibitory processes in the brain might contribute to some of the perceptual problems in autism. And I know this is something when you're talking about neural discoordination, it's different, but how, if all, might that be related or tied in, or are these two completely unrelated phenomena?
So that's a great question. Thank you for asking that. The actual features that we're studying, the coordinating features, are very intimately tied to interneuron function and the appropriate, I would go so far as not to merely call it balance, but coordination of excitation and inhibition. At any one moment, every 100 or so milliseconds, inhibition changes rather radically in the hippocampus, and so does excitation. And how they change in lockstep with each other, either over rigidly, as in the case of the Fragile X knockout, or not in a sufficiently reliable way, as in other models, for example, after models that involve schizophrenia kinds of features. These kinds of coordinating events are what we're trying to study, and as we proceed right now, we're starting to look at individual interneuron subtypes, and we have various models. I glossed over them without talking about them, but various models of how the dynamics of this interaction between excitation and inhibition, and in particular the plasticity of inhibition, is related to these phenomena. So it all emerges, if you will, from a interplay between excitation and inhibition, but it's not a static phenomenon that there's just more or less. It's changing very dynamically on fast and multiple time scales.
I see. Okay, that's really helpful. That's interesting. One of the listeners asked, does development, age, or experience change how the neural responses, how this coordination operates as far as the timing goes? Is there any kind of developmental arc or curve to this?
There almost surely is, I have to say. We embarrassingly have not studied it properly across development. No one properly has. We're just beginning such a study in a different model, mouse, or rather rat, to look at them in adolescence as well as in adulthood. These studies are obviously fairly challenging, but I think it really requires looking across development. And one of the interesting opportunities as we've developed, for example, these mini microscope recording technologies, we'll be able to look in the same individual animal as it develops across periods of order a month or so, to see how these changes may or may not occur. And they're very clear predictions about what kinds of coordinating events will occur, will be important, and which will be broken down in several of these particular models. But that's work that's ripe for doing now.
Okay, that would be interesting to see. So thanks for that. And I think we'll have to go ahead and end it there. Our conference room is filling up with a meeting that'll happen after us. So I just want to say, for those of you that tuned in and maybe missed portions of today's broadcast, we'll have a full replay of today's presentation online. You can also share that with colleagues. We'll go on a December break next month, and we'll be back with a webinar session on Spectrum in January, I think late January. So check spectrumnews.org for that full schedule. I want to thank everyone for tuning in, and especially thank Professor Fenton for giving us a great presentation. And until next time, thanks everybody.
Thanks everyone.